Good afternoon. Why don't we go ahead and get seated and we'll get started. My name is Alan Rudolph. I'm the Vice President for Research here at Colorado State, and I want to be the first to welcome you this evening to a forum that we are hosting on coronavirus outbreak and some of the things that we're doing here in the research enterprise. So this is very much focused on the research enterprise at CSU and the work that we've done historically in infectious diseases, but now in our current situation focused on the crisis of the coronavirus outbreak. And I'd like to start with some opening remarks before I turn it over to Mark Zabel, and I'll introduce Mark and the panel and let the forum continue. And I'd like to start with reading a news passage. The city had slowed to a near halt. Schools were closed, church services were banned. The city began shutting down. The government staggered its hours to limit crowding on the streets and on streetcars. Commissioners overseeing the district's closed schools, along with playgrounds, theaters, vaudeville houses, and places of amusement. In a list of 12 rules to prevent the disease's spread, the Surgeon General wrote that people should, quote, avoid needless crowding and, quote, wash your hands before eating. One slogan was, quote, cover up each cough and sneeze. If you don't, you'll spread the disease, unquote. Those who were healthy wore masks when venturing outside. People who were known to be infected were threatened with a fine if they were seen in public. These are not the words from people in China where the current outbreak of coronavirus, or now known as COVID-2019, where 75,000 cases of the new coronavirus and 2,000 deaths have been recorded as of this morning. But these are quotes from over 100 years ago by Bill Sardo, one of the last remaining survivors of the Spanish flu outbreak in 1918. That outbreak killed 600,000 people in the United States and 50 to 100 million people worldwide. More people that were killed in that virus than any war that the Earth has experienced. Here we are 100 years later, facing an increased frequency of outbreaks in both animals and humans, and asking ourselves with each one, quote, how prepared are we? What innovations can we apply to do better? And will this evolve as we know each one of these could be a global pandemic? The good news is we're doing better in each one of these events, but it's still a reminder that we're not prepared to face a fast-spreading disease that could repeat the horrific events of past pandemics. We live in a future world and a current world of reduced effectiveness of antibiotics. We still have a slow development time for new vaccines and countermeasures. And China faces an outbreak in humans at the heels of its worst animal outbreak, having lost estimates of 50% of their pig population to a disease called African swine fever just in the last year. I've been engaged in investing in infectious disease outbreaks for most of my career, starting at DARPA over 20 years ago when I met Ken Alabek, who was the second in command in the former Soviet Union, who defected to tell the world of the need for global biodefense programs based on their use of infectious disease as weapons. This is a new feature of unfortunate outbreaks that not only are they endemic or could flare up naturally, but we have adversaries who will use them against us. Over the course of my career, I've invested $1.5 billion in this problem of increasing outbreaks and trying to increase resiliency in industry, government, and now in academia. Over the course of that time, I've seen a lot of research and development, and part of the reason I came to CSU is for the reasons you are about to hear. I've encountered no other ecosystem like CSU's that has as much to contribute in this problem set as we do. From fundamental research to commercial production of countermeasures, across the spectrum, CSU excels with its ideas, its partnerships we can create, and engagement with communities from our region to around the globe. So please enjoy tonight's event because you're very fortunate to hear world-leading experts on infectious diseases from a discovery point of view all the way to translating those ideas to scale and practices that will affect Colorado and affect the society as we know it across the globe. So with that, I'd like to introduce tonight's sort of MC, and Mark Zabel, who's really been instrumental with Ellen Fisher in our office, and Lauren Clam, and Patty Nash, a bunch of the folks who have put together tonight's event. But Mark really comes to this event with a lot of his own experience in an infectious disease based on proteins, prions, which won the Nobel Prize in '96. Very unusual disease outbreak. You may remember manifest as diseases we called mad cow or sheep scrapie or chronic wasting's disease in deer and elk. Mark is currently the Associate Research Dean for the College of Veterinary Medicine and Biomedical Sciences. He received his postdoctoral training in prions, the protein that is involved with that infection, in the laboratory of Dr. Adriano Aguzzi at the Neuropath Institute in Zurich. He was the first American to join an international lab, and that's another example. These pathogens know no boundaries. They can be at our doors in 24 hours or less. And so there's a lot of international engagement that's part of this story, and Mark's background reflects that. He's coordinated international courses on his topic, and I think another feature of CSU's work in this area is a very strong international engagement. And Mark also coordinated these curricula in the European Molecular Biology Organization while at the Institute. So I'm going to let Mark come up, introduce the panel, and then we'll get started with various aspects of the topic on the coronavirus outbreak, and I want to thank you for coming out tonight.
Well, good evening, everyone. Thanks for coming, and thanks for your interest in this current outbreak of a virus, the family of which has been around for a while, decades, but now we have a new flavor, and I'm glad that you're here. You know, as Alan said, Colorado State has a long, rich history in infectious disease research. But as a land-grant institution, I do feel that CSU has a responsibility, a commitment to educating and serving our constituents, Coloradans. And so I'm pleased that you all could make it, and I just want to thank our panel tonight for being here and sharing their expertise on this topic. Before we begin, and before I introduce the panel, I just want to remind everyone, I mean, you guys all look like fine, respectful individuals, but I know that sometimes topics like these can bring out some emotions and some passions, and I just want to remind everyone, you know, that we're going to respect everyone's questions and perspectives, and we're here tonight to focus on the science and the research and the data behind it, okay? And so that's what our function is to just talk about the facts, hopefully provide some clarity, some perspective, perhaps even some guidance depending on the questions that you have. And when we do open it up, you know, we have a lot of different perspectives up here on the stage and also in the audience that hopefully we can answer your questions, okay? So I'd like to start out then by introducing our panel up here on the stage. So starting with Brian Geiss. Brian is the associate professor in my department, Microbiology, Immunology and Pathology, and has studied viral replication, antiviral development and diagnostic assay development at Colorado State University for over 14 years. Dr. Geiss works closely in a very interdisciplinary way, and I'm sure he'll talk about that tonight, with a team of virologists, analytic chemists, and engineers at CSU to develop new technologies that can detect ultra-low levels of virus and viral pathogenesis in patient samples and in aerosol samples. Dr. Rebekah Kading established her obtained her BS in entomology and wildlife conservation from the University of Delaware, a master's degree in entomology from the University of Arkansas, PhD in molecular microbiology and immunology from the Johns Hopkins Bloomberg School of Public Health between 2007 and 2014. Dr. Kading led studies on the ecology of arthropod-borne viruses in Colorado, Uganda, and Guatemala, and the transmission of Rift Valley fever virus by mosquitoes at the CDC Division of Vector-Borne Diseases. Her research program at CSU is currently focused on Rift Valley fever transmission, as well as virus circulation among bats in Uganda. Dr. Kading's research in Uganda is investigating the potential for spillover of infectious agents from bats to people, which may be relevant for tonight's talk. She serves the scientific community as an editorial advisor for the journal PLOS, Public Library of Science, Neglected Tropical Diseases, and a steering committee member for the Bat One Health Research Network. Dr. Tony Schountz is an associate professor in the Department of Microbiology, Immunology, and Pathology. His research interests lie in virus reservoir-host relationships, particularly those of bats and their viruses. His current funded projects include work on Middle East respiratory syndrome coronavirus and bat influenza viruses. And as an immunologist, he's an excellent immunologist too. It's not listed on here, but. And then finally, we have Dr. Lorann Stallones. She graduated from the University of Texas School of Public Health in Houston with an MPH in International Health in 1975. She served as a health planner in the Peace Corps in Palau. She received a PhD in epidemiology from the University of Texas School of Public Health in Houston in 1982 and serves as the director of the graduate degree program in public health served until recently at the CSU School of Public Health and the Colorado School of Public Health from 2007 to 2019, and currently is staff supervisor for our One Health Institute. Dr. Stallones was the principal investigator on an NIH Fogarty International Center-funded project which trained more than 100 Chinese scholars about agricultural injury prevention research. She is co-author of a textbook entitled Global Health, Ecological Foundations from Oxford University Press. And I do also want to point out just a few of our expert audience members. We have Dr. Candice Mathiasen. She is the director of the Infectious Disease Research and Response Network. We have Dr. Bob Ellis, who is CSU's biosafety officer. And we have Dr. Angela Bosco-Louth, who is an assistant professor in the Department of Biomedical Sciences in the College of Veterinary Medicine and Biomedical Sciences. So I'm very confident that we have a great panel that can answer any questions you might have, again, based on the sound science, the research, and the data that we have currently. So with that, I think what we're going to do is have each panel member speak for a few minutes about their expertise and what they'd like you to know. And then once we do that, then we'll open up the floor for questions and we'll get you some answers, okay? So, Brian?
Can you hear me? No? All right. Oh, is it on?
It's on now.
Okay. Hi. So as Mark said, my name is Brian Geiss. So I'm going to try not to spit on it. So as Mark said, my name is Brian Geiss. I'm an associate professor in the microbiology department, and I've been here at CSU for 14 years. My main research focus over that time has really been trying to understand how viruses replicate and then figure out ways to use that information to defeat those viruses in the form of developing new antivirals that can help treat patients in the clinic, as well as trying to develop new types of diagnostic assays so that we can detect virus even sooner than we can with the clinically available assays that your physician might actually be using, or that the public health department is currently having in use. I come at this from a very, as I mentioned, translational standpoint. I view viruses as a threat to our public health, but the fact that we can use science to actually understand how they work and use that information to then develop new therapeutics and new tools to detect them as the most a very powerful tool. So to give you a little bit of background of where I come from in terms of the work I've done, for about 10 years I did most of my work on trying to identify new types of antiviral drugs that can affect viruses somewhat similar to coronaviruses, such as West Nile virus, dengue virus, chikungunya virus, which are not in the same family as the coronavirus that we're talking about today, but have somewhat similar types of genomes. And trying to develop new antivirals that can interfere with the processes that those viruses use in cells to replicate themselves. And so what we do is we actually identify key points in the virus's replication machinery that they use to make more of themselves, and figure out how to break those points, figure out how to find small molecule drugs that we can put into patients eventually that can interfere with those processes and then block the ability of the virus to replicate, giving your own immune system a chance to actually fight off that infection and then give you long-term immunity. And so we do this in a number of different ways, primarily by understanding the molecular mechanisms these viruses actually use to replicate themselves. And I won't go into too much of the weeds, but my philosophy is if you don't know how a machine works, you can't figure out how to break it. And that's what we try to do. We figure out how the machine works, and we figure out where to put the monkey wrench in to break it. So that's the first part. That's what I spent about 10 years working on. So I'm fairly familiar with the antiviral development and use that's going on with the coronavirus outbreak right now. The other aspect of our research is to try to develop new tools to detect infection and do it in a way that's faster than is currently available with current clinical diagnostics. So currently, if you get infected by a virus such as coronavirus or any other type of pathogen, your physician would take a small sample from you and then send that to a clinical diagnostic laboratory where they would extract viral genomes from that sample and then try to detect that pathogen using a technique like PCR, trying to basically detect its nucleic acid. And that works really well, and that's what's currently being used to detect the coronavirus outbreak. The downside of that is that it does take a while to actually get those results, and during that time, people are still getting sick. So what I've been doing over the last 5 years is trying to develop new tools in collaboration with analytical chemists such as Chuck Henry here at CSU, as well as engineers such as Tom Chen in electrical engineering and David Dandy in chemical and biological engineering, to develop these new tools where we can detect infection even sooner than the current PCR assays that are being used, and eventually trying to use those so that you can detect infection at the point of care. So if you go to the physician's office, being able to find out in a shorter period of time whether you're infected or not, so that your physician can give you actionable information. And so that's a thumbnail sketch of the work that I do. And I can answer questions about the diagnostics as well as the antiviral clinical trials that are going on right now.
Good evening. My name is Rebekah Kading, and I'm an assistant professor in microbiology, immunology, and pathology. And my background is in entomology and wildlife conservation, and so my lab deals with questions that regarding the circulation of pathogens in nature and how they're transmitted to people and the efficiency with which that happens. And so I've worked on a variety of different disease systems over the years. My dissertation work was on malaria in Zambia, and then I did a postdoctoral fellowship and spent some time after that at the Centers for Disease Control. And so we studied West Nile virus here in Colorado, as well as in Guatemala, to compare temperate and tropical transmission cycles. And so the perspective that I'm coming at this, the coronavirus work, is that some of the work we were doing in Uganda was involving the ecology and circulation of viruses in bats, and the potential role of bats as reservoirs for viruses. And the original focus was for arboviruses in particular. But over the course of our studies, we started interacting with some communities in caves in eastern Uganda that were experiencing high human traffic, as far as for local population as well as some tourism. And it was really interesting ecologically too, as far as the bats that were present in those caves and not much understood about them. And so the research that we're just beginning to undertake, and so we've had our kickoff meeting and we'll be traveling back there this spring to actually start the field sampling, will be to try to understand more about viruses, and coronaviruses was on our to-do list anyway, because of the bat species that are present there. And so we want to understand more about the circulation of viruses at that human-bat interface, understanding that bats are reservoirs to some pathogens, in particular coronaviruses. And so we want to understand how those viruses would get from bats to people. What are the points of vulnerability that might happen, as far as are the people handling bats, or are they just stirring up some aerosols, or what is that contact level that would result in a spillover event? And is there a seasonal component to that? What are the various aspects of risk that people face when they come into contact with the natural reservoir for some of these pathogens? And so we're also looking at this very much from a One Health perspective, and so the health of people, animals, and the environment are very closely knit together. And when we talk about bats, we also have to recognize that bats are very diverse and ecologically important species, a group of species. And so they portray a lot of important ecological roles. They're keystone species, and so they're involved with pollination, seed dispersal, insect consumption, and so there's a lot of ecological services that they're providing that we have to recognize. And so when we approach research questions that involve human pathogens, we're asking these as far as how can we protect public health, but also conserve bats and the role that they're carrying out in the environment. And so we'll be looking at the dispersal patterns of bats, their movement patterns, how these movement patterns are associated with the shedding of viruses into the environment. Is there a seasonal component to that, and how the people are interacting with the bats that could potentially result in pathogen spillover. Thank you.
Okay, I'm Tony Schountz, and like Doctors Geiss and Kading, I'm also in the Microbiology, Immunology, and Pathology department. And I've focused on the reservoirs of these viruses. And so for those of you that aren't familiar with what a reservoir is, it's a particular species of animal, sometimes there could be 2 or 3 species of animal, in which these viruses circulate, but they don't cause any disease. And so for whatever reason, they've sort of come to an evolutionary détente with one another that if you don't bug me, I won't bug you. And that's perfectly fine if you're a virus, because a virus doesn't want to kill the animal it's in, because if it does, it loses its home, right? So what happens is we end up getting what we call spillover events, when the virus jumps from, let's say in this case, a bat into a humans, and now suddenly this virus, which has evolved to be highly optimized to exist peacefully in a certain species, is now in this brand new environment in which it has never seen before. And as bad as it is for the people, most people will survive the infection and overcome it. But it's terrible for the virus, because it's gonna die. Either the patient dies and so does the virus, or the patient mounts an immune response and clears the infection, right? So the virus doesn't want to get into people any more than we want it to get into us. So what I want to understand is, how do these viruses infect their reservoir hosts without causing disease? And if you think about it, these animal hosts, bats for example, they've already figured out how to handle this new coronavirus, which is clearly a bat virus. MERS coronavirus, Ebola viruses in all likelihood, Marburg virus, Nipah virus. Anybody seen that movie Contagion? Who saw Contagion? So I don't want to give the plot or the end away. But to me, that's the scariest virus on earth in terms of potential disease consequence. And the one that I've worked with in my past, not here at CSU because we don't have the proper containment facilities for that one, but at the National Institutes of Health. So when I started my career, my sort of independent research career, it was focused on hantaviruses. Who's heard of those? Anybody from Colorado should know about hantaviruses, right? And the reservoir, does anybody know the reservoir host of the hantavirus that we have around here? What's the animal that hosts it? It's like my classroom. The deer mouse, right? It's called Paramiscus maniculatus. And likewise, we can infect deer mice with the virus, that hantavirus, and they get infected. You look at them, they look perfectly fine. Nothing bugs them, right? So what is it about their ability to cope with that virus that when that virus gets in humans, it's 70% fatal without medical intervention, and even with medical intervention, it's about 35% fatal. So those are the kinds of questions that I'm interested in, is what do these rodents and bats know about their viruses that we could learn from and perhaps develop some novel clinical strategies that might mimic their responses such that we could then control or mitigate disease progression in humans, right? So I got into bats back in 2005 or 6, whenever SARS, the first SARS, not this current SARS virus, they're both SARS-like viruses, but they're behaving quite differently. And it was discovered by a group in China as well as another group in Hong Kong, which of course is part of China. But they independently discovered that these viruses are commonly found in bats. And so that's what precipitated my transition from hantaviruses into bat-borne viruses. I still do hantavirus work. We're working on some therapeutics for that. But the bat-borne viruses became very interesting to me because they hosted all of these really notorious viruses, right? I've already mentioned a few of them. And they do so seemingly without disease. Now, I want to be perfectly clear about something. These viruses are very species-specific in terms of their reservoir hosts, right? There are about 5,000 species of mammals, and about 1,200 of those species are bats, right? So there's a large number of bats. They're the second largest mammalian group behind the rodents at about 1,800 species. So 2/3 of all mammals on this planet are either a bat or a rodent. Does that make sense? And if you look at the really significant human pathogens that spill over from these animals, it's either a bat virus or a rodent virus. There are certainly others. We're not going to get into the mosquito viruses because they are really kind of scary to me. But nonetheless, those are the viruses that I think about on a regular basis because they are hosted by mammals, yet when they get into another mammal, humans, they can cause this rather dramatic and nasty disease. So consequently, that's my interest, is understanding how these bats can cope with these viruses and how have the bats shaped the virus evolution, right? Because if you're a virus and you get into a new animal, you either got to figure out a way to establish that sort of immunological détente. You don't bug me, I won't bug you. Or the host is either going to mount an immune response and clear it, or the host is going to die, right? And that speciation event inside the new host animal, in this case bats, clearly shapes how that virus is going to behave. And so bats have some really unusual features about their immune systems that we think are probably instrumental in shaping how these viruses have evolved and why they may cause substantial disease in humans. And that's the focus of my laboratory, including MERS coronavirus. We're going in and we're looking at mutations in the virus to see how that changes the immunological outcome and the infectious outcome. Because if we can identify certain genes in the MERS coronavirus genome that are essential for it to cause disease, maybe we can go in and just delete those genes. And now we have a virus that looks to your immune system like the regular MERS coronavirus, but it can't cause disease. And now we have a vaccine candidate. And I suspect that this is going to be something that will be examined with this new SARS coronavirus as well. Okay.
So I don't have a laboratory, and this is not my primary area of research. I'm an epidemiologist by training, so I look at patterns of disease in human populations, and we look at these things by analogy to other patterns of disease. So all of these things are fascinating to me. For those of you who want to know a day-by-day update of what's going on as far as we know, with the best information, the World Health Organization is publishing a daily report. It's called a situation analysis. You can just Google WHO and you can say coronavirus And now it's COVID-19, and you can get right to the page, and it's got the actual number of cases that are currently known. It's got the cases by location. It's got epidemic curves of the human cases that have occurred outside of China. But right now, what we do know is over 90%, almost 98% of all of the cases that have been reported to date have occurred in China. And a majority of those actually occurred in Hubei province, which to me is near and dear to my heart. As you heard, I spent many years working with colleagues in China, and Wuhan was the city where we worked most. And I have been in touch with some of my colleagues there who've been isolated in their homes. And one of my colleagues' husband is a physician, so he's been working this outbreak since the beginning. And I've got to say that it's quite reassuring when you compare this coronavirus to SARS and also to MERS. The case fatality rate for SARS that's been reported was 9.6%. The case fatality rate for MERS was 29.9%. These two outbreaks were not very readily transmitted human to human. What we're seeing with this coronavirus is clearly it's learned to jump from human to human very effectively. But in China right now, the reported case fatality rate is 2.9% in Hubei, and it's about 2.3% throughout the rest of China. But for those cases that have been reported outside of China, right now it's 0.3% fatality rate. So this is not as fatal an infection as some of the previous coronaviruses that we've been dealing with. I think that we created an amazing and disturbing natural experiment by isolating the people on the cruise ship and leaving them there, because the vast majority of cases that have occurred outside of China occurred on that cruise ship. And the attack rate for that group is 1.22 per 0.9 per thousand. The attack rate in Hubei was 1 per 1,000, and in the rest of China, it's only been 0.05 per thousand. So we created a natural experiment on how well this virus can be transmitted in a contained environment. We do know that cruise ships, ships in general, with recirculating air, are really an incubator for respiratory diseases. And so that's really all I have to say.
Great. Thank you, Lorann, and all the panelists. Just want to acknowledge and appreciate and thank the panelists for staying on time. We're actually a little ahead of time. It's amazing what you can do when you don't allow them to do PowerPoint presentations. Their timeliness is your gain. At this point, I think I'd like to throw the panel open for questions, if anybody has any. Do we have any runners? Oh, great. Thanks.
I think I have a couple of questions here. One for the epidemiologist is, why do entities avoid getting the information out in a timely manner, like the poor ophthalmologist who died after sounding the alarm, and then he was, visited by the police and told to cease and desist. And so, secondly, since you brought up the cruise ship, it seemed to be acting as an incubator. And so CDC is recommending, negative airflow or HEPA filter. So should we have a sick person? I know a lot of the people that came off the planes that were transported from China were being put in military facilities. What would be your recommendation for housing ill patients if such were the case? And here's another question, probably for the virologist, is as in the Ebola transformation, we were able to create some pretty effective medication based on the survivors that came. So do you think we might be able to do that with the current people who have survived this little incident.
So I think your first question was what happened to the ophthalmologist, and I think that this is it's unfolding. The stories are unfolding now. So I think the best you can do is follow what the reports are from the credible sources to really try to understand what happened. And I really have no information that I can relate in regard to that. I am not a physician, and I don't deal with treating patients, but I think that what you have when you have the containment facilities with the proper airflow and exchange and control, you're going to reduce the transmission of the virus. And you have to understand that on a cruise ship, you don't have any of those things in place. It's just an environment that's a normal air exchange environment. So that's why it becomes an incubator. And that's why people are put in isolation if they come off with signs and symptoms, or if they're in quarantine to protect just until they find out whether or not they're positive for the disease. So I think that the control measures that are in place are appropriate, and I think they're doing the best they can to make sure there's no further transmission of the disease.
So I'll take the second one, and that is hospital containment. I don't know the details, but I'm certain the state health department is on this. The CDC has certainly issued guidelines, and I imagine most hospitals have infection control protocols for such a scenario. They've probably rehearsed them over and over until they're saying, why are we doing this? It's not going to happen. And now here we are, it's happening. The third question about therapeutic approaches, I think what you're alluding to with the Ebola outbreak was the monoclonal antibodies. And I believe Dr. Rudolph knows quite a bit about this because I believe DARPA was involved in that. Is that correct? And would you like to talk about that?
So the use of monoclonal antibodies, as Tony said, was the Ebola rescue for the 3 patients that came to the University of Nebraska. You're also referring to what's been used in other outbreaks as convalescent plasma. So from people who do recover, there are, just like Tony was referring to, if you've developed resistance or you recover, how you do that is very interesting to research. And so there have been attempts, and there are some even here at CSU, to look at convalescent plasma from those organisms, mostly animals that have recovered from these diseases. So there are things you can learn from people who develop a resistance or who recover similarly with animals. So that the convalescent plasma is sort of the term of art that you can search on to look for what kind of history has there been around investing in those kinds of approaches. Thanks for the question.
Thank you. I'm Jim Fountain. I'm a grad student here. I have a question on your data you're using for your research, data protocols, data protection. So in my research, as I go to school here, I'm really concerned about events that make the obtaining of the research data susceptible to cybersecurity or manipulation so that it's not really as valid or as accurate as it could be. So given the coronavirus, so the closed nature of the Chinese society, the need for WHO and CDC to grab data that's accurate, effective, and timely. Are you aware of, in your research, any special things you've now done to make sure the data protocols are followed, that your data is accurate and valid, or that WHO and CDC are doing to get the data they need, and maybe are doing some bending of the rules that puts them at risk for manipulation just because they have to work around the Chinese way of governing to get the data you need to do the research. But in the end, it's data protection. So that's where my question goes. So it's to anybody, really.
So does somebody want to comment on?
Well, this is easy for me. I have no data yet, so I don't have anything to. So we're all relying upon our IT security people to make sure there are no intrusions, but they get intrusions all the time, right? You're probably aware of this, and they have to put up these blocks in place. Other than that, we really haven't started on any of this yet, so we don't have anything to risk yet.
So I'll just follow up with that real quickly to say that next generation sequencing technology has really revolutionized how we respond to these outbreaks as well. And I think within weeks we had a rough draft of that sequence, and we're able to start sort of aligning it to other known coronaviruses, and determined that it was probably of mammalian origin as the vector, and that's how we arrived at bats. And so once that data starts to come out, it's really difficult for any government or institution to suppress that, right? And so I think what you saw there during this outbreak was a transformation of how rapidly we can start to understand these viruses and put them in the context of where they might have evolved from. And so you know, I would just, again, want to kind of focus on the science. Governmental organizations are one thing we don't control, but there are a lot of good Chinese researchers, and I'm sure Lorann could attest to that, that are doing incredible work, and even some have sacrificed their lives to get that data out.
Yeah. Hi, my name is Mary Marshall. I'd just like to ask Brian Geiss about your point of care non-PCR diagnostics. What's it based on? And then there's, you know, recent studies have shown that the timing of this, taking the swabs, it makes a difference in detection. And between the oral swabs and anal swabs, and I wonder if you have a comment on that, that, you know, if it's oral, it's earlier, and the anal is later, but then more effective.
All right, thank you, Mary, for your question. So basically, what we're trying to do is develop a diagnostic assay where we can take a salivary sample from a patient and put that on a very inexpensive electrochemical device. It's basically a fine gold wire that is coated with viral antigen. So some proteins from the virus that we've made in bacteria. There's no biosafety issues with it at all. But we can use it to actually detect the antibody that's secreted into the saliva that during the infection. And the value of this assay is that we've actually shown in other viral systems that we can detect infection almost immediately after it occurs. The earliest time point we tested with a Zika virus diagnostic that we're working on is 4 days after infection. So to give you an idea of how that relates to normal infection or clinical diagnostics, it might take 7 to 10 days to actually detect those type of samples. So it's currently in research and development. We're not at the point where we're going to put these assays out on the market within the next month or even year. But what we're trying to do right now is develop some reagents, some materials that we can use to actually detect these coronavirus proteins or antibodies that are present in patient samples. So Dr. Schountz and I are working on a project together to detect some of the viral antigens and antibodies towards those antigens in these samples, as well as we're working with people at Washington University School of Medicine to develop those as well. So to get to your second question in terms of oral swab versus anal, there is actually data suggesting that the oral virus particles and viral antigen and antibodies in saliva can be found fairly early after infection. There has been some reports, though they're still very sparse because it's an unfolding epidemic or unfolding process, that there might be some viral genomes in fecal samples. But how infectious they are, we don't know. And very likely those would come out later during the infection process, because this is more likely than not a respiratory infection that goes in through the upper GI tract. Does that answer your question? Okay, great.
Hi guys, just wanted to say thank you before I begin. I read an article today released by the Washington Post. I don't recall like the author of it, but I remember them reporting something about like an estimate of like 1 year until a vaccine is kind of found and confirmed. And I just wanted to know your guys' thoughts on like an estimate on when a vaccine will come about or get created.
Our resident immunologist.
I'll take that. So the fully honest and truthful answer is I have no idea. So the coronaviruses are a little challenging when it comes to vaccine development. In fact, a number of viruses have been shown to be very challenging for vaccine development. My suspicion is that this outbreak will come and go before a vaccine comes to market, but I don't know that for sure. You know, the dynamics of this infection is quite different than MERS and SARS. So SARS came and went in about 9 months. MERS emerged in 2012 and is still ongoing. And no one has come up with a vaccine for MERS that is really useful or even can be used in humans at all. And here we are 8 years later. So I don't think history is on our side. So I hate to be a downer about it, but that's kind of my perspective on that. Now, it could be that this virus is a little more tractable in terms of vaccine development because every one of these viruses is a little bit different, and some are easier and some are harder than others. So I know there are a lot of companies that say they're going to have one soon, but I'll believe it when I see it. So I'm naturally skeptical about that.
Hi. I was just wondering from each of you whether you think this is an overreaction or not.
Let's start with Rebekah. She had a question.
An overreaction. Well, I think that it's, this is the third human pathogenic coronavirus that's emerged that seems to have a bat origin. And so, I think that's something to learn from. So we've been through SARS and MERS and now this one. And so I think, it is, time to take a step and consider this pattern and what we can learn from it and what we need to change to prevent it from happening in the future.
So, if I may, so following that up, so this particular virus, right, is it an overreaction? And I don't know. So I'm more worried about influenza, quite frankly, than I am this virus. But I know how flu is going to behave every year. I have a pretty good idea. And I also know there are vaccines that aren't the greatest, but they can certainly reduce your chance of getting disease. And even if you do get disease, the vaccine will probably limit it so it's not as severe and perhaps shorten your illness and shorten the amount of time that you're actually transmitting it so that you have a lower probability of transmitting it. We also have a number of really effective drugs for influenza, right? We don't have those for this virus because it's new. And so following up what Dr. Kading said, is it an overreaction for this virus? I don't know. For coronaviruses? No, I don't think so. I think we're going to see more in the future. 2.5 years ago, there was a group from the Wuhan Institute of Virology where the virus, Wuhan is the city where it was first detected. They presented data here in Fort Collins at a conference we hosted that showed that there are a lot of these SARS-like coronaviruses in bats, dozens of them. We don't know if they can cause disease, but there's a lot of these coronaviruses out there. And so I think coronavirus outbreaks are going to be an ongoing issue that we can deal with. They probably were in the past as well, but we just weren't smart enough or had the tools or whatever to really effectively follow and determine that these viruses were coming out and causing disease outbreaks. And so we've only talked about 3 human pathogens, right? SARS, and then MERS, and then now SARS-2, which is the virus. The disease is called COVID-19 or something like that, but the virus is called SARS-2. We didn't talk about SADS coronavirus, which is a swine virus that was discovered about 3 years ago that was also a coronavirus from a bat that killed a huge number of piglets in China, and they had to destroy a larger number of piglets simply to interrupt the transmission. So I think it's not an overreaction in terms of coronaviruses. Whether it is for this particular coronavirus, I don't really have an opinion one way or the other, but we're still in the early stages of this disease and this virus.
Oh, Lorann, I'd like you want to comment?
I would say that from a public health point of view, we tend to look at two things. One is the number of people that are impacted by whatever the disease is, irregardless of whether it's fatal or whether it's highly fatal. And given the numbers of cases that we've seen already of this disease, and we don't know how many more we're going to see, I think my initial thought when I read about it was maybe we're overreacting a bit. On the other hand, I think maybe not. If you want to reduce the number of cases, you need to get, and with a respiratory disease, you need to get control early. And so I think it's probably not an overreaction from that perspective.
Heather, thanks for your patience.
Lory.
Just one more from Brian.
All right.
Thanks, Heather.
I would actually agree with everything my colleague said. At this point, it's an unfolding story. We are seeing a lot of cases and a lot of information in the news about what's going on, and I think that is unfortunately making people more fearful of this than maybe they need to be. We encounter infectious diseases all the time. Influenza, as Dr. Schountz said, is much more of a concern to me personally than this particular coronavirus at this point. However, that being said, this is the third coronavirus that we've seen in the last 20 or so years that can cause human disease. So what I think it does do is point to this family of viruses as something that we need to have, more on our radar and invest more time and effort into understanding how they work and understanding their epidemiology and how they pass in populations so that if another one comes out in the future, we'll be even more prepared.
Thank you. Heather?
Heather Pidcock, Chief Medical Research Officer. I just wanted to respond to that question as well by pointing out that even though this virus kills fewer people per case, it infects a lot more people, so it has already killed more than MERS and SARS together. So, and I think that we've been very lucky. Even though there have been more and more outbreaks of pathogens in the recent decades, we've been lucky that we haven't had the combination of a very lethal pathogen that has a long incubation time. Once you, and that is easy to spread. I think these are the sort of sentinel events that tell us that that is a high risk and that we need to address it. So I don't think it's an overreaction at all.
Thank you, Heather.
I was just wondering if you could talk about how a virus, whether it be influenza or this one, peaks. Like I've heard, seasonal flu has already peaked, and then eventually how it goes away, or does it ever go away?
That's a great question because I think we've been sort of talking around that issue about how virulent it can be, how fatal it can be, and how transmissible it can be. So I will open it up to the expert virologist. How about Brian?
Well,
I'll take it. Okay. So the idea of peaking is, where a virus infects as many people as it will, and then starts to filter out of the population because no more people are either getting infected or the herd immunity is enough to actually control that virus in the overall population. So at this point, we don't have a great idea of what that number actually is for this coronavirus. How long it will persist in China and in the various parts of the world that it's being spreading in right now, we're not quite sure. The containment efforts that are underway in a lot of the parts of the world will help mitigate that to some extent. But at this point, we don't really have a great idea how far it will spread and how long it will spread. Now, I will say that because there appears to be a large amount of human-to-human transmission with this, potentially through aerosol, though it hasn't been absolutely proven that it's an aerosol transmission, close proximity to people does seem to increase transmission rates. So when you have areas of very high population density, such as Wuhan, which has about 12 million people, then the potential for transmission actually goes up. Whereas if you get into areas of the world that have a lower population density, the transmission probability actually will be lower. And so it might not spread as fast. So in terms of how it was spread over time, that makes a huge difference.
Rebekah, would you like to?
Oh, we were just having a sidebar about the concept of R naught, and so which describes the reproductive potential for a virus. So the number of new infections resulting from a single infection, and so this is expressed in as the R naught. And so the R naught for this virus, I think, is just over 2. And so on average, there's just over 2 additional cases for each initial case. To put that into context, the R naught for measles is 18. So a very highly transmissible virus. I think Ebola was around 3 or 4. So again, so this is something that would be about 2 or 3 cases for each one.
So another important aspect, Dr. Geiss alluded to population density. Another one is population susceptibility, right? So this is a completely novel virus, and nobody has any pre-existing immunity to it. And so everybody is susceptible, right? So you combine that with high population density and a virus that in all likelihood is transmitted via the respiratory route, and I think probably through diarrhea and things like that, then you have a much more efficient or a higher risk of transmission. So if you look at influenza, though, if we immunize enough people, then you lower the susceptible population, right? And by doing that, you can interrupt the transmission cycle. So another issue that we have to think about is with influenza or measles, when somebody gets infected with those viruses, they don't feel ill for many days afterwards, but yet they're still producing and shedding the virus from themselves. And because they don't feel sick, they go about doing their daily business to go to work and hang out with their friends, and all the while they're transmitting. There's a suggestion that this virus, this coronavirus, behaves in a similar way, but I haven't seen any convincing evidence of that. We have what's been reported out of China, but it sure fits that profile that people probably transmit for a few days prior to disease onset. So those are the kinds of people that worry us. But the ones that really worry us are those that get infected and never show signs of disease, yet they still transmit the infection, right? Because they go around and they spread and they don't know they're spreading. West Nile viruses, for example, 80% of people who get infected with West Nile virus never know they were infected. The good thing about West Nile, though, is that it's not transmitted other than by mosquito transmission, right? And the virus doesn't replicate in humans very well at all. So these are all these factors that sort of conspire to determine how efficiently a microbe is going to pass through a population, right? So if we have a vaccine, we should all use that vaccine, even though we may not want to get it, like for influenza, because it's terrible, right? Because we can really knock influenza down quite a bit if everybody were to get vaccinated. Unfortunately, we have a very low level of vaccination rates in this country for influenza.
That's a great point. I just wanted to follow up. I think that kind of goes back to Dr. Pitcoat's comment about incubation time. And to put it in another perspective, I think perhaps the virus that exemplifies that incubation period and how it affects transmission is HIV, right? Because you can be asymptomatic for years and not realize it. So, yeah. So, question here.
Thanks, everybody. Michael Schmidt. I work in molecular medicine, and I have a couple of questions. One would be, have you seen or has anybody published any patterns in the host characteristics in terms of who is responding more severely to the viral infection, et cetera? And then the second question would really be a little bit related. So from the standpoint of diagnosis, across the world, are we using similar diagnostic methods when we say we have a confirmed case? Is everybody using a similar method to confirm that diagnosis?
Great question.
So on the host characteristics, the information that's available, there's some on the World Health Organization website. they're suggesting that older people and younger people, but there aren't a lot of details yet that I've seen where you really have the demographics and you feel secure that you know what you're really looking at.
Sort of, go ahead, Brian.
So the diagnostic test that has been approved by CDC is a PCR for 2 different targets of the viral genome. One of them is the nucleoprotein gene. And the main reason the nucleoprotein is typically used is because by far and away, the most messenger RNA is produced off of that gene, because the nucleoprotein is instrumental in not only viral genome replication, but also viral packaging. They typically have activities that suppress the cell's ability to defend itself. And so consequently, there seems to be an abundance of that particular messenger RNA. And so consequently, it makes it a very attractive target for this diagnostic PCR assay.
And related to that, the sequence for the coronavirus that we're talking about today actually was provided to the entire global or scientific community in mid-January. And so we know exactly what the sequence is. And using that sequence, the CDC was able to develop a very robust diagnostic assay that is very similar to what's being used in China as well as in Japan and Korea. The details are going to differ a little bit between their target selection, but we're using the same type of assay all across the world, predominantly because the sequence of the genome of the virus is very simple, and we can make assays for that very easily. Some of the more complicated assays, such as looking for antibody against the virus that you're producing during an immune response, those type of assays are still not available yet, simply because we don't have the materials available yet to do that. So we have to make proteins to actually develop these assays that are used for serology, and that takes time and effort. And so there are people around the country and around the world who are actually developing those materials to supplement these nucleic acid assays that are being developed and the CDC is using to help give confirmation of infection.
I don't think, oh yes, I have 2 questions. I think you partially answered one of them is, what difference does it make if you've been provided with a live virus as opposed to the sequencing? Because I know in Australia had the live virus from the patient, and I think they felt that they made more progress because of that. But and then I just have another question about, you've talked about the reservoirs, but what about intermediate hosts? Do you want to talk about pangolins? And then just this morning, ProMed moderator was suggesting that maybe cats should be tested, especially, pets that belong to people who are infected or people who are infected coming into contact with pets, and perhaps he suggested that this should be studied in China with in the cat population.
So what is it about bats that harbor so many of these different viruses?
If I knew that answer, I would be retired. I'll probably work until I die trying to figure this one out. So your first question about companion animals perhaps being susceptible, I saw that in ProMed this morning. I didn't read it, though. It was something to do with some veterinary association.
I sent it to them. I could send it to you.
Okay, well, if it's in ProMed, then I have it. So I don't know the answer to that. In terms of what makes bats different, there are a lot of people that think they're, I hate this word, special. Oh, go ahead.
Talking about the intermediate hosts like pangolins.
Oh, okay. So that's all speculation too, and that's based on sequence that was detected in a pangolin some months ago that has a great deal of sequence similarity to this. Could that be a route? Of course it could be, but there's no experimental evidence to say that that's correct. So when you have one pangolin that has a sequence in it that looks like part of this virus, that's suggestive, but until you follow it up with experimental work with pangolins, infect them with the virus and see if they're susceptible and can they shed it, it's just sort of an observation that has no substance behind it. We have the saying in science, when your sample size is n, and when n equals 1, it might as well equal 0, because it doesn't really tell you much, right? Does that make sense? I'm not trying to dodge it. I don't really know. I'm telling you the truth.
So maybe a follow-up question for that would be, you know, bats are the only flying mammal. Does that contribute to their ability to have more zoonotic potential, and the fact that they can fly to distances, greater distances, and perhaps contact individuals more readily?
So we're going to be here all night. So there are some who think that when bats, because bats fly, they generate a lot of body heat, and that's true. And they say, well, this is like a fever, and they're going to, that's why they can host these viruses, because they have a fever all the time when they fly. But from a physiological perspective, a fever response is far more complicated than just you out running around and generating body heat because you ran 15 miles, you know, backwards on one leg. The fever response is preceded by a number of immune proteins that are made during an infection. In fact, the fever is because of those proteins. So by the time fever occurs, your immune response is already engaging the pathogen. So personally, I think that there's zero connection and there's zero experimental evidence to that. So we did an experiment where we took some bat cells and we incubated them at normal temperature with Ebola virus. That wasn't done here, by the way. We don't have those facilities for Ebola here. This was at NIH. And then we raised the temperature of the cells to mimic what would be the temperature, the body temperature of a bat. And we measured how much Ebola was coming out of those infected cells. And it was exactly the same. So that's the only experimental evidence that looks at this. And as poor as it is, it's better than what everybody else has done. So I'll just leave it at that. So there are some other features about bats, though, that make them unusual. Some of them seem to have their antiviral systems always on at an elevated level. So we humans and the mice that we use, which, you know, when we teach immunology or virology, we usually talk about humans and mice. Those are two mammals out of the 5,000 species of mammals, and then we try to draw inferences, and that's a big mistake in my view. So these bats have this system, this antiviral system, that instead of being off like humans and mice, it's on at this very modest level. And they never make any inflammatory response. I've never seen an inflammatory response in a bat, what I would say is a bona fide inflammatory response. So if you're a virus and you evolve in that environment where your antiviral defenses are always at this level, when you inadvertently get into a human being where the antiviral system is off, the virus is going to have free reign. It's like, well, where are all these antiviral proteins? They're gone. They're not here. And so the virus replicates like mad. This is a hypothesis that we have, but we have no experimental evidence to support that. So I think it's more about how the biology of the bats has shaped the viruses that have made them so dangerous that when they get into humans, they cause these diseases, right? But I don't want to distract from the simple observation that although these bat viruses are notorious, right? Ebola and Marburg and SARS and MERS and, you know, Nipah and Hendra viruses, there are a lot more of these viruses that kill people that come from rodents than from bats, right? I can name 15 hantaviruses that kill people, and those are all from rodents. So anyway, I think the bats shape these viruses, but whether or not that has an impact on human disease is just speculation on my part at this time. So,
Thanks, Tony. We have a question over here.
Yes, thank you, and thanks for this great information. I was wondering, I'd heard in the province where this originated that 5 million people just kind of left the area. That seems to be a concern. I wondered if they know or tried to contain that, what's happened to that, and the fact that the CDC wants to go over there and study it and have access to the areas, but China said, no, we're going to keep you kind of corralled. Do you know if that's changed any?
So again, I don't know that we have enough information about those geopolitical issues to speculate, so I would caution against doing that. And I don't think we have the expertise on this panel to address that. So I thank you for your question, but I think we have to just stick to the science.
I wonder if, from the past Zika virus infections, the difficulties that low-income countries with not very good laboratory infrastructure struggle with confirmation of diagnosis. Is there something in place right now to increase the possibility for confirmation of diagnosis in these countries, in other countries?
So I think that's where a WHO intervention would be required. And I think what would happen is they're monitoring for outbreaks, and if it reaches a certain threshold, or if there's a potential mathematical formula that they use that says, we should look in this particular city because of this number of cases and the infrastructure or lack thereof that they have for not just diagnostics, but also for medical treatment, then they may deploy a team, but they can't just go. They have to be invited by the government, and I don't want to get into the government part of it, so.
Yeah, again, look at the situation reports. On February 14th, WHO deployed the testing kits to 56 countries. So they are in the logistics group from WHO is actively engaged in making the testing kits accessible in all over the world.
Couple of questions again. In an effort to be proactive and just, curiosity research, I asked, should we encounter this virus, what would we use to disinfect or kill it? And I was told, use the viricide. Well, So I called the manufacturer. The manufacturer said, thank you for calling. Don't use our product. As a matter of fact, no manufacturer can tell you right now that their product will kill that virus because we need research time with it, and we need to know what exactly will do that. So with that said, could we use a bleach, and what would that dilution be? And secondly, for example, if we have a medical here at CSU, we're quite fortunate. We have firefighters respond, we have paramedics, and we have police. So it's a great response. But that said, our first responders perhaps need some training to know, what are they dealing with and what should they do. And so, again, it seems like a preventive sort of thing to do, because our first responders lots of times, they're on the front line and they see these things before we start to pick up a pattern, or it's confirmed. So 2 questions. Thank you.
Lorann, do you want to speak to the public health?
Yeah, I think what I would suggest is if you want to know what the recommendations are, the Centers for Disease Control has some flyers that are one-pagers that really give people the guidance as best they know how to manage these things. And so that's really the best source at this time, I think.
Maybe I'd just add quickly that, you don't necessarily need a viricide to reduce the virus levels on your hands and your face, or, and I think CDC, those pamphlets that Lorann was referring to, they say wash your hands, right? That may not kill the virus, but it does remove it from your hands and send it down the sink, and then it will eventually die outside of a host. Oh, so surface decontamination. Yeah, okay. Oh, yes, our expert audience member, Biosafety Officer Bob Ellis.
Well, I'll talk all around that. But basically, the answer that you got from the manufacturer was absolutely correct, because FDA hasn't ever approved anything for this particular virus. But there are coronacidal antimicrobials or antivirals or decontaminating agents that are out there. And if it says on the label that it's going to kill coronavirus, this is a coronavirus. So it depends on if you're looking for something that will actually kill the coronavirus or this particular virus. And until somebody does that, we can't say that it's going to kill this absolute particular virus. Now, as far as the ambulances and fire departments, we've worked with them over many years, and we had a live exercise, mock exercise, about 3 months ago, just to go again through the exercise. What if in one of our biosafety level 3 laboratories we had an incident where we had a medical emergency and we needed to get that person out as a life-saving event? And so we worked hand in hand with the fire department, police department, the ambulances all we come with the fire department. This wasn't the first time we've done this. We've done this over the last 25, 30 years from time to time. And it basically boils down to the PPE that they wear, they meaning the emergency responders, and they're well-versed, well-trained with that. They will then clean out the ambulance that they've used, any other equipment that they've used, and they will make sure that that is deconned before it's used again. We've gone over that scenario with a lot of different microbial agents and different crews. So is it going to be perfect? Maybe not, but I really think it's going to be close to it. Would a what? I think a bleach solution would work, and by a bleach solution, the ones that we use most frequently, and we try not to use them any more than we have to, because bleach solutions will corrode and start to decay equipment, and especially stainless steel. So we try to stay away from those in our laboratories. But bleach solution, if you look on the label, most bleaches are 5.25% hypochlorite. Hypochlorite is what is in the bleach that's actually going to be the antimicrobial. And if you take 1 part of that and add it to 9 parts water and use it right then, it should be effective against almost most things. Maybe not spores. Viruses don't produce spores. But you don't make that dilution and then keep it around for a month and then use it. You use it fresh.
And I just want to be clear, he's talking about general virucides. He mentioned that this is not a recommendation for this virus because the work hasn't been done. But your question, and the reason I thought it was important to stand up and add to the conversation, was around first response. And one of the features of land-grant universities, of course, is the extension network. And CSU has a very prominent historical extension network. And most recently, the first responders in that network have organized themselves around a unit called EDEN, the Emergency Disaster Extension Network. So in every county in the state of Colorado, Colorado State has an extension touch or person, and they are getting more and more involved in this EDEN network, where the extension system is using the same knowledge creation you're hearing here, but also from CDC and the official channels, to communicate with our region, our state, people in concerns in rural communities, commodity groups that have concerns about diseases affecting their business or things like that. So the university, by its very nature of being a land grant and having this extension system, has a touch, including the first responders, and they're all hazards. So the same folks who are now responding to fire and flood are getting the training around infectious disease, and Reagan Adams is here. She's our veterinary extension specialist. She's a veterinarian, very much focused on transmitting that knowledge into our communities. So we're very much focused on what you're talking about, which is not only training of our first responders, but making sure that CSU is able to transmit that knowledge out into the community. And she's also very involved in the kind of modeling of that you've heard about training or mock training kind of activities. So thanks for the question.
And I'd add to that too, that in our biosafety office, we were one of 5 different universities that had NIH grants to train emergency responders, and we did that in not right here in Fort Collins. We tried to get that really organized with the emergency responders, and it just turned out it never happened. But we were part of teams that went into New Mexico for customs training those people that would be taking people in with unknown substances possibly. A couple of us went out to Washington, D.C. We trained over 1,500 firefighters out there, helped train those. And this was part of a consortium with Duke University, University of Louisville, University of Chicago, and us to get that done. Unfortunately, the grant only lasted for a couple of years, but we've definitely got the expertise and the ability to do that. And one question while I'm up here that hasn't been asked, so I will ask it, is why do we want to do the research here? Why do we think we can handle that research? And should we be doing it here? And the answers to that are all affirmative. We've got the infrastructure, we have the facilities, we have the expertise of anybody to do that research here. And we have the protocols in place, essentially, that we've already worked with viruses like this. We've already done a huge amount of biosafety level 3 research. We have the experience, and who else would do it better? I don't think anybody would. And if I thought that we couldn't do it as biosafety director, I would definitely say we're not doing that here until we can get to that point. We're there.
Thank you, Bob. So I think in the interest of time, if there's one more question, we'll take it. If not, or even if so, I want to reserve just a few minutes at the end for our director of our Infectious Disease Research and Response Network, Dr. Mathiasen. So I think Alan set that up very nicely about response and extension. So if there is one more question anyone has? If not, oh, yep, great.
It seems like they know the geographic origin of the virus, but do they actually know patient 0 and how it was transmitted to patient 0? And are they attacking it from that point?
So the original information that was coming out suggested it started in this wet market in Wuhan, China. But I think it was the New England Journal of Medicine paper where they found a serum sample that had antibodies to this virus, and that serum sample was collected like 2 weeks prior to the outbreak in that market. And it usually takes 2 or 3 weeks to get a really robust antibody response occurring. So I think many people will think that it probably started at least mid-November, if not sooner. But of course, they didn't identify who that patient was on that December 1st blood draw. I'm sure the Chinese know who that person is, and I'm sure they're trying to drill back in time to see how that individual may have acquired it. But right now, it looks like that person could have been involved in the early transmission event, but we don't know because we don't know how far back it goes. So.
Thank you, Tony. All right, great questions, everyone. So before we finish, I just want to give Dr. Mathiasen just a few minutes to speak about IDRRN.
Thank you. I'm Candace Mathiasen, and my area of expertise is in pathobiology. I study chronic wasting disease in deer and elk and moose. And one of the things that I do as well is be director over the Infectious Disease Research and Response Network. This is one of the programs of research and scholarly excellence that was born out of Dr. Rudolph's office. The VPR's office has provided funding for these groups. There are about 22 of them, I believe, on campus now. We are one of those. And the members that you see up on the panel are individuals that are part of this network as well. So the work that they do individually, as well as work that we are doing to try to move across campus and bring a interdisciplinary research program to these questions, including that of the recent coronavirus outbreak. And so I just wanted to give you a little bit of an idea of what this looks like, in that we have team leaders for each of these groups that we have ongoing right now. And the teams themselves are comprised of individuals from microbiology, immunology, and pathology, throughout the veterinary school itself, across the College of Liberal Arts. And let's see, we also have atmospheric sciences involved in these groups. And the idea here is to bring various perspectives to the question to be able to address a broader impact of that. So this first one that you see here is that associated with companion animal cognitive disorders and individuals that are working in this area. These groups are meant to be fluid so that individual expertise from people within these groups can move across boundaries and give their expertise to other questions. A second group that's ongoing right now is trying to use mass spec for immunological analysis, and you can see that Dr. Schountz is on this team as well. A third group is interested in trying to better understand the prion protein. So I mentioned that I study chronic wasting disease, which is a disease that's the prion disease of elk, deer, and moose, and now reindeer as well. And so what we really don't know is what is the function of this protein. And it looks like it may have some impact in cancer as well. And so there's a group of individuals that is recently growing to include, again, similar folks across campus asking these questions and developing a better understanding as well as diagnostics and therapeutics for these diseases. And then, of course, our topic this evening is that of the coronavirus, and a group that is just recently forming the last couple weeks, individuals coming into this team, as well as our panelists, to try to address this question from a broader perspective. So thank you.
Thank you, Candy. Yeah, looks like that team is so new and emerging. We're still looking for a leader. Panelists. All right. Well, thank you so much for showing up. You know, the Fort Collins community never ceases to amaze me about how smart you are and how engaged you are and how concerned you are. And so thank you for showing up. And also, I want to thank my colleagues from my department. I hope that we've been able to convey sort of the collegiality and the synergy that, that we strive to work together to make these discoveries and push these and translate them to benefit all of us. And so if we could just give a round of applause for our panelists. That'd be great. And just one quick plug, if you haven't gotten enough of these panel discussions, I and a couple of graduate students of mine host a community-based outreach event called Science on Tap. That occurs next Monday at the Wolverine Letterpress and Public House. We're going to have another panel with Dr. Dick Bowen, Dr. Danielle Adney, and Dr. Charlie Kalischer, who will also be fielding questions and giving their perspective on the novel coronavirus outbreak. So you're welcome to attend that as well. Once again, thanks. And one quick thing, I saw that our president, Dr. McConnell, snuck in to attend too, and I just wanted to say thank you for doing that, and I hope you're as impressed as I am with the great audience. So thank you all again. Safe travels home tonight.